Every minute, millions of white blood cells, the body's immune soldiers, patrol the circulatory vessels looking for signs of infection or injury. When pathogens invade the body, they attack its healthy tissues. At the first sign of infection, the immune cells gear into action, beginning their journey by identifying specific molecules serving as stop signs. Each molecule has its own exit password, which signals a different immune cell, essentially a highly effective call to arms. The cells roll to a halt, begin to flatten on the blood vessel wall, and then squeeze through on their way to neighboring tissues, where they seek out and destroy invading pathogens. Unfortunately, this system is exploited by cancerous cells, which migrate in the blood, metastasizing in distant parts of the body, the dreaded spread of cancer. Much like a Trojan horse, cancer cells are able to identify and respond to these stop sign passwords, thereby gaining access to neighboring tissues, as we're about to see. Dr. Ronen Alon has recently gained insider's information about these so-called stop sign molecules. What are we looking at, Doctor?
Would you like to see?
Thank you.
Well, you're looking at immune cells traveling through blood vessel, just like this vessel.
Right. So what we're seeing here, in fact, is like a glimpse into the body.
This is really a chamber which is meant to simulate the blood flow.
Yeah, how do you do that?
We're taking components that we can take out from human tissues, build up these site of action from scratch, and see how all this interplay really happens.
How do chemokines recruit immune cells to action? Using their experimental model, Alon and his team were able to actually see this multi-step process for the first time. They discovered a surprising fact. The chemokine signal is not enough for immune cells to squeeze through vessel walls. There's another part to the puzzle.
The blood flow. Because as of now, the idea was that chemicals that are injected or that are produced during inflammation, getting to be displayed in the vessel wall, are sufficient. And the blood flow was just the default state in the system. We, by having this reconstitution system that we developed here, we could take out that component and then bring it in. Once we've done these manipulations, we actually found out that blood flow is not just there, but it's actually required.
Dr. Alon and his team have added another angle. Blood flow, they have found, not only filters the immune cells, but also triggers the next stage in the process, when the immune cells squeeze their way through the vessel walls on their way to fighting infection.
That squeezing is one of the most mysterious processes in modern immunology, and we are actually trying to simulate the vessel wall architecture.
This study may contribute to future therapies that fine-tune migration processes, enhancing beneficial immune responses while blocking the spread of cancer.